Hello and welcome back to the 15th lecture on BioMEMS, biomicroelectromechanical systems. Let's do a quick preview of what had been done in the previous lecture. We talked about extensively the designing of an electrokinetic pumping network, and then started developing or discussing some initial concepts of biology, elementary level concepts, which are important for the understanding of the diagnostics, especially clinical diagnostics technology. And so essentially we started with the cell. We discussed what the basic unit of life, that is the cell, comprises of. Essentially it is a gelatinous membrane which actually contains a liquid called cytoplasm, and there are several organelles like the Golgi bodies, the energy warehouses for the cell, the mitochondria, the endoplasmic reticulum, the protein warehouse, so on and so forth inside, floating around in this cytoplasm. And in the center there is a distinct organelle called the nucleus, which contains an area called, or region called chromatin, which essentially has a bunch of supercoiled, highly compressed DNA molecules in the form of double Y shapes or chromosomes. We also discussed that the human body has almost all cells which has 46 chromosomes except the reproductive ones, which has 23, and they combine on fusion to develop 46 essentially. And we discussed some facts regarding sizes and scales of the species. For example, if you open up a chromosome and try to lay it out on a flat surface, a stretched DNA which comes out of a chromosome is approximately 6 centimeters long, okay, and contains as high as about 510 to the power 6 base pairs or molecules inside it. So we then started discussing about the elementary DNA structure and talked at length about the Watson Crick model, which is like a double helical, a twisted ladder kind of a structure. We discussed some issues regarding the nomenclature which is used for understanding the direction of placement of the sugar phosphate backbone. So there are 5 carbons in the sugar ring, numbered 1 to 5. The third carbon, there is a phosphodiester linkage which joins one sugar molecule to the next in the backbone, and on the fifth carbon there is on the fourth carbon there is a linkage, the phosphodiester linkage, which goes all the way to the to another sugar atom. So this is the way the sugar atoms are all linked together, and the first carbon on every sugar is also bonded to a base, and there are 2 such complementary base pairs, A and T, adenine and thymine, cytosine and guanine. So let's look at how these base pairs are clubbed in a structure. So this right here really is the DNA backbone made up of sugar and phosphate, and the molecules as illustrated here are the base pairs. So as you see here, this is a thymine T, and this is an adenine A, okay. Similarly this is cytosine and guanine. These are all chemically classified into pyrimidines, and this adenine and guanine are chemically classified as purines, okay. And one more interesting factor is if you see the linkage between these base pairs, the thymine and adenine are connected using 2 hydrogen bonds here and here, whereas the cytosine and guanine are connected using 3, okay. So crisscrossing is not permissible because it creates a thermodynamically unfavorable situation. 3 bonds, if clubbed together essentially with 2 active sites, would leave 1 bond which is dangling, or vice versa. So therefore a cytosine can never bond to an adenine, or a thymine can never bond to a guanine. It is only paired up in this unique manner so that it's a minimum energy configuration. So there is a huge negative charge on the backbone. This phosphodiester linkage between the 2 sugar molecules, as we illustrated in the last lecture, is essentially having a PO4 minus, and the more the number of sugar molecules, the more the number of the phosphodiester linkages, and higher is the negativity of the DNA, or the negative charge of the DNA. So if you put this in a solution, there is a tendency of attraction between the different moieties including the base pairs, and therefore the DNA really coils up, okay, and bundles together, and it remains in that form until and unless it is forced through maybe a set of sieves using electric field, external electric field, etc. So you can see here in the figure below, we talk about the actual structure. The double helical coil here is coiled, as you see. It goes in this twisted ladder kind of fashion, and the connecting base pairs in between keep the strands integral and attached to each other and bonded to each other. And so therefore, the whole DNA molecule is nothing but a bunch of sequences of these different base pairs, and essentially the complementary is defined by what sequence this base pair here has. And so therefore DNA has really a very important advantage of kind of self assembling if you know one strand or one sequence in this particular molecule. Let us talk about some other aspects of DNA which is important for detection and diagnostics. What is DNA hybridization? So essentially, when a DNA is heated to about more than 90 degrees Celsius, or even exposed to a pH of less than 12 or greater than 12, there is a tendency of these complementary strands to kind of split apart along the hydrogen bond. So there is a cleavage between the different hydrogen bonds in between the base pairs, okay, along these directions. If you look at these are the hydrogen bonds really, and there would be a tendency of cleaving the molecule away from each other by cleaving these hydrogen bonds. So such cleaving can occur when you are subjecting the DNA either to a kinetic energy, which means that essentially you are providing free vibrations to the different strands which are bonded together through the base pairs by the hydrogen bonds, okay. So they go above a certain frequency of vibration and the strands separate. And another way is to put it in a pH solution, an extremely basic pH solution, for it to kind of split apart, okay. So therefore this is also known as DNA denaturation. This process of DNA splitting apart is also known as DNA denaturation. This process, however, is reversible, okay. So therefore once it happens on an exposure to a melting temperature of Tm greater than, let's say, 65 degrees Celsius, but again less than 90 degrees, would result in the two complementary single stranded DNA structures so formulated joining back together, okay. So this we know as hybridization. Hybridization is essentially the joining of two single stranded DNA molecules on a certain melting temperature, which is about 60 degrees or a little more, for the molecule to be again integral back to normal. So there is a development of hydrogen bonds between the different base pairs, and the strands typically join together to form a double stranded DNA molecule. So this is what hybridization essentially is, okay. Further, hybridization can happen between any two complementary moieties like a DNA and a DNA, a DNA and an RNA. The difference between RNA and DNA essentially is that RNA is a single stranded structure, and the thymine group in an RNA is replaced with another base called uracil. Essentially that is what the primary difference is between an RNA and a DNA. So the hybridization can take place between two DNAs, DNA and an RNA, or an RNA and an RNA, and essentially the only thing which is needed for the hybridization to happen or realize is the complementarity of the two joining strands, okay. So this really can provide a very sensitive means of detection, this hybridization protocol, and it can detect specific nucleotide sequences. So essentially, if you know what you are trying to detect, okay, or if you at least have a hunch of what you are trying to detect, you could really design an exact complementary sequence and try to hybridize your target with the complementary sequence. And if so, there is a in this process there is a hybridization which happens, and there is effectively some kind of an immobilization which is taking place because of this hybridization. You can say that the target had the same sequence or the complementary sequence as the sequence that you designed it for, okay. So the flank which is there to capture is also known as the capture probe, and the other portion which kind of comes and binds to this capture probe is the target DNA. So it's a very sensitive, very specific means of detecting sequences on nucleotide, and not so extremely rapid. So there are several factors which affect this hybridization process: a variation in temperature, salt and buffer concentrations, and essentially the guanine cytosine content are some of the things which would really vary this hybridization property. Logically that makes sense because more is the number of cytosine and guanine, more is the triple hydrogen bonds. Okay, so the two base pairs guanine and cytosine, as you saw before, join with 3 hydrogen bonds. In this particular illustration here, it joins with 3 hydrogen bonds. So therefore, more is the GC content, or more is the guanine cytosine content in the DNA, more is the bondage essentially between both these strands, and so hybridization can be a little more difficult, and the melting temperature or the melting characteristic may change because of the presence of an extra hydrogen bond in a more abundant manner in such a molecular sequence. And therefore, temperature, salt and buffer concentration and also the GC content can really be critical for finding out the hybridization rate and also the melting characteristics of the particular target molecule. One more issue here is that the rate of hybridization has been found to be proportional to the concentration of the target and probe. Okay, and it is essentially limited by the concentration of the lower material or the lower abundant material. The one which has a lower concentration is the limiting concentration. So therefore, hybridization can be characterized or is effectively proportional to the concentrations of both the species which are binding the target molecule as well as the capture probe. And the limitation is imposed by the species with the lower concentration. If there is more number of targets than the capture probe, hybridization will be limited by the capture probes, or if there is more number of capture probes than the target, then hybridization essentially will be limited by the target. So there won't be any binding after a while because target is unavailable. So that's what this process of hybridization is. So if you really look at some of these kind of relative absorbance trends at 260 nanometers, you can see that the way that DNA would kind of go from its native state into the denatured state and then again get renatured does change the relative absorbance of light at 260 nanometers. So this is the denatured case where you are just heating it, and as you see here with the rise in temperature, there is a sudden change in the rate of rise of the relative absorbance around 90 degrees, which is also effectively the denaturation temperature. Okay, in the case of renatured DNA, there is there is no absorption or almost a very limited absorption up till the temperature reaches let's say about 80 degrees or so, beyond which there is a sudden rise in the relative absorbance. Okay, so the way that the absorbance to light behaves for these different processes is also to an extent different. For a native DNA, this would happen somewhere around again about 90 degrees or so, that the absorption criteria would change, but it would happen quite towards the end of the spectrum, almost around 80 degrees or so, there would be a sudden rise in the absorption. So you can actually have differential characteristics, as you can see here, of optical absorbance, okay, between these 3 states: denatured, renatured and native state of hybridized DNA. So these characterizations are important for essentially determining the hybridization process. You have to understand here that the DNA molecule being extremely small, about 20 to 30 nanometers Armstrongs in radius and about a few microns in length, you have to rely on the optical data or alike data as given here in this illustration for finding out the signatures, and that can give some information about the state that the DNA is in essentially. So for DNA hybridization arrays, it is critical that these characterizations are done in advance, and the calibrations so obtained can be compared to the binding characteristics later on with any unknown target and set of capture probes to determine what the target can possibly be in terms of sequences. And there are certain other issues that I would like to discuss here about hybridization. So hybridization essentially is not a foolproof process. There may be illustrations where there is a stringent binding, like for example here, if you look at this particular figure here, you have almost complementary base pairs opposed to each other, like for example C and G are binding, A and T are binding here, again T and A are binding, this is CG, this is GC, this is CG, this is a G and a T. Okay, so they don't bind. So essentially this is something that is there in the molecule by virtue of its presence, where it doesn't bind. So here there essentially will not be any bond which is developed. Similarly, the A and T here would again be something which is binding. So here there would be a bond. Okay, let me just erase this for convenience. So there is no bond in the GT case. You have another A and T here where there would be a bond. So essentially, as you are seeing here, most of the base pairs facing each other are complementary, and they would do the binding, whereas there is only one base pair here which is non-complementary, and there won't be any characteristic binding in this particular region. The guanine to thymine binding does not exist. Okay, so therefore still it is a very, very stringent, very perfect stitching process or hybridization process, as opposed to this other case here on the top, which shows about a reduced stringency case, where as you see there are a lot of base mismatches. So there is a A and a G in this region which is a mismatch, there is a C and C in this region which is again a mismatch, again a C and C, and again an A and G. So there are so many mismatches in such a particular situation. The DNA still binds because the number of bonds which are holding it together are more probably, and the number of bonds which are not getting formulated because of non-complementary base pairs, okay, facing each other. But again, the forces given by the binding pairs are probably much more, so that the DNA can still be held together. But the problem here is that it is not that stringently bonded to both the flanks are not very stringently bonded to each other, as in the case of the highly stringent case, hybridization. So it is desirable that, especially for the readout aspect as well, when you are trying to decipher what is there on a target by looking at the capture probe, that we go for a stringent hybridization case where almost 99% of the molecules do bind to each other because they are exactly complementary of each other. Okay, so therefore this is something which is very important in designing hybridization arrays, and the focus of the target of the capture probe really should be able to have a highly stringent hybridization process for the sake of clarity and false readouts. Okay, so there is another kind of very interesting phenomena with the DNA which was invented way back in about 1985, and this is known as the polymerase chain reaction. Okay, so essentially there is also a process which was invented by a scientist by the name of Rob Mullis. Okay, and he was essentially awarded the Nobel Prize for this discovery. This reaction is really something which is able to give a breakthrough, I would say, in the field of molecular biology and identification, especially in diagnostics. We'll look at what this reaction is, but before starting it, let me just reiterate some concepts of sensing mechanisms and sensors. So one of the interesting aspects of a sensor is how to amplify a weak signal. Okay, so when we are talking about a certain sequence of bases in a DNA, it is essentially a bunch of maybe a few molecules, and if we are attaching normal probes or we are actually, by probes what I mean is fluorescent probes, okay, or we are attaching a detection mechanism which is very normalized without really very high sensitivity, then there is really a problem of identification of the signal. Okay, and so therefore it is important to enhance the signal so that we really get a very detailed, a very good data for confirming whether a DNA with a certain base pair sequence is present on a sample of interest. And so it is almost in the interest of proper sensing to develop amplification protocols for the signal. One way of amplifying would be just to take the photons generated by something like fluorescence using a probe, and then trying to kind of collect this, amplify this, and go ahead with the amplified signal. Another way could be that if I can somehow chemically copy the base pair sequence in vitro, that means outside, in pretty much similar manner as it happens within the cells, then that can be a great advantage, because essentially we are copying a certain size or a certain region of the DNA, a sequence which is unique to a particular organism or a particular cell of interest. And therefore the idea would be that you take a particular sequence and try to chemically use certain steps so that we can exactly copy identical sequences and make multiple copies, and that way the signal can be increased. So that's what the PCR reaction is. If you look into the way that cells replicate and divide, essentially this is what happens, that the DNA zips down or zips open, and then there is a replication of the individual daughter strands, which actually get double the number, and they are exactly split into two daughter cells during the cell division process. So the same chemistry or the same mechanism can be used in vitro as well. Okay, and so Robert Mullis was the first guy to kind of propose this PCR or polymerase chain reaction. So the issues which are important here are following. One is that the amplification process is driven by an enzyme called DNA polymerase, and there are also identification sequences called primers, which are short single stranded DNA strands, and you put that in the solution at the very beginning before starting the chemical reaction. I will in just about a minute show a schematic which would give you a better understanding of what these primers are and what they do really. Okay, and some characteristics of a PCR process is that billions of copies of DNA molecules can be made within about hours in the laboratory, and essentially it is very useful in the research, diagnosis, forensics, where large samples are required from small concentrations. Another interesting, and this I forgot to mention, is the area of forensics. During investigation protocols, from just blood stains of a patient or the person who has actually conducted the crime, it is important from that little stain to decipher some information about the fingerprinting aspect of the person. Okay, it gives an idea of who the person is essentially, and so if you look at that aspect, PCR is essentially immensely vital, because how do you take a stain which is probably there for the last few hours after any such criminal event has happened, and how do you detect from that small amount of sample which is dried the fingerprints of essentially the people who were there on the site of the crime, or about the identity of the person who has suffered or who is the victim. Okay, so forensic science really relies a lot on PCR as well, apart from diagnostic science, for this technique. So let us look at what this interesting technique is really, and essentially it is a 3-step thermal cycling process which does the following. So in the first step, if you look at, so again I would just like to reiterate, this is what a DNA structure really looks like. It is like a double helical structure with base pairs which are connected in a complementary manner to each other, right. So let us think that this is that double helical structure here, which is kept in the solution with a bunch of different other chemicals. So the first step that a PCR reaction would execute is that you basically denature the DNA by heating it for about 1 minute or so to a temperature of about more than 90 degrees Celsius. So essentially you are breaking open the hydrogen bonds, and you are not doing that at the cost of breaking the sugar phosphate backbone. You are just simply splitting the two hydrogen bonds or the two base pairs apart, and in the process separating the strands, with the strands individually still remaining intact but getting separated from each other. That is denaturation. So the first step is denaturation, and the second step, you design these intelligent identification sequences called primers. Okay, and there are two sequences here, one which starts and binds at the forward direction, another in the reverse flank of the other gene of, or the other flank of interest, or the other strand of the DNA, and essentially this gives you an identity or an idea about the base pair sequence of the DNA. So the PCR is an effective process because without looking anyway into this particular flank, and by just developing some identification complementary strands, 20 base pairs, 30 base pairs, maybe about 50 base pairs long, which kind of binds to the sequence which are there in both flanks of this DNA, one in the, let's say, one strand and another in the complementary strand in the reverse direction, we are done. Okay, so this identification is good enough for giving us an idea of what the sequences are on a particular DNA molecule. So the primers again kind of go and anneal to the daughter strands which have been formulated by denaturation, and that happens at a certain temperature, and this temperature is about 54 degrees Celsius. Another very interesting factor here is that why is it needed to have two set of primers, forward and reverse. And what would happen really if we start doing this synthesis from the same flank on both ends of the DNA molecule. Okay. So this is a very interesting aspect, that if there are two complementary strands just opposite to each other, the primers which are exact complementary of each other would bind to each other rather than getting bound to the strand. So therefore you have to have a forward and a reverse sequence. And why both sequences are needed is that you have to really copy both the molecules. Okay. The idea is to be able to exactly duplicate or copy into two folds the number of molecules that are originally present within the solution. So this is the annealing step. So once the primer binds, you have other moieties or other chemical species within the chemical reaction or the parent solution which does the remaining job. Okay. And the species which are there include an enzyme which is also called Taq polymerase. There are these individual sequences called DNTPs, or essentially nucleotides. And what a nucleotide is really is one of these base pairs, okay, A, T, C or G, bonded to the first carbon of this sugar molecule and a part of this phosphate. So essentially the base pair, the sugar and the phosphate all together, this is essentially what a nucleotide means. So there are 4 nucleotides with this base pair varying between adenine, thymine, cytosine and guanine. So there is a sugar phosphate thymine, there is a sugar phosphate cytosine, there is a sugar phosphate adenine, and then there is a sugar phosphate guanine. These 4 base pairs are present in the solution. And then there are these enzymes which would essentially be intelligent molecules, biomolecules, which would look at what is present on the DNA flank to be sutured. Okay. So if it finds out that here we have an A, the enzyme would try to pick up a T molecule and affix this here, and not only that, it also tries to suture the phosphate linkage to the earlier sugar which has already been sutured here. Okay. So not only the enzyme picks up the base pair, particular base pair, the phosphate, sugar and the base pair, and sutures it to the complementary base pair on the DNA, but also sutures the phosphate connecting both the sugars. The other sugar is fixed and the sugar is which is mobile is on the particular DNTP, okay, or the nucleotide. So that way you can actually get a whole coverage in a very rapid manner, and the enzyme can suture quickly by adding the base pair, joining it to the daughter strand and kind of bridging whatever has been left open and zipping up the DNA again. But mind you, this new DNA molecule has been made from molecules individually present in the solution by help of the enzyme. Okay. The Taq polymerase also goes up to an extent of doing proofreading. So if there is some by mistake, if there is something which has been wrongly sutured, or there is not a good enough match between the base pair which is there on the daughter strand and the particular DNTP that has been picked up. So the Taq polymerase always has this tendency of kind of proofreading and cleaving back the incompleted or the wrongly sutured base pair back into the solution. Okay. So it is also a self proofreading mechanism, and it is a very stringent process because errors happen while copying DNA inside cells at the rate of about 1 in 10 to the power 9 such events. So it is such a highly stringent, such a highly accurate error free process, and if it were to be a little more intensive, we will have a lot more mutations, and there won't be a stability in the way we have evolved over the past. So just because of the sturdiness of the DNA molecule, we have been able to evolve in a manner with minimum possible mutations, because of this error rate while copying is extremely low. It's about 1 in 10 to the power 9 such suturing events. So however, once the enzyme kind of copies the DNA, it zips up and makes 2 strands out of the 1 strand that had been, or 1 DNA, double stranded DNA that had been opened up here or denatured here. So now if you repeat this cycle for n times, you should ideally get about 2 to the power n copies of the parent number of molecules which were there in the solution, right? And this is exactly the replica of one another. They are exactly copying the sequence that was there on the parent molecule, and thus essentially you have amplified the chemical signal in terms of creating identical molecules throughout the particular chemical reaction process. So now let us look at some of the other aspects. So essentially you have here you see a double stranded structure, you are splitting it open and bonding it to primers, and then you are completing it through an enzyme, and these are on different temperature scales, 90 degrees, 55 degrees, 70 degrees Celsius, and then from here again you do repeat the cycle by heating it and denaturing, bonding it to the primers again, and then trying to extend it. So you have essentially again another denature, anneal and extend step, and this continues, and as you see here there is a slow growth in the number of sequences, and these are all the chemical signals which are amplified products made from substitute DNA out of the template which was given by the original DNA. So it essentially contains information about the sequence and exactly copy the sequence in the following manner. Okay. So some take homes here are that primers essentially are short strands of nucleotides which are complementary to specific regions of a target DNA which is to be amplified. Hence the end sequence of short regions of the target DNA to be copied is needed, and therefore, so now how it is important in diagnostics is again a very critical question. Okay. So if I am able to somehow sequence a particular area, a definitive area in a certain region of the gene of a cell of interest, and if that particular region is able to address a particular entity or a cell of interest. Okay. So I know now that there is a database where we have a sequence of the DNA, and we also have the name of the moiety which contains the DNA. So it is addressing the different entities. So you have, let us say, a certain bacterial cell which has a certain region of the DNA which is unique to that particular cell. If you have another cell Y, you have another region of the DNA or another sequence somewhere in the DNA which is very unique to Y. So it addresses the X, Y, so on, etc. Now the whole idea is that if I want to explore a particular sample for a target of interest, I want to find out whether, let us say in a food sample I have a certain bacteria X. Okay. So how do I do that? So I essentially try to read if there is a sequence which was addressing X or which has been found out to address X in that particular sample, and for doing that you develop primers which can go and attach to the end flanks, one in the forward, one in the reverse direction, and copy or start copying the DNA. So if I put such primers there with exact complementariness on the first 20 sequences, maybe on either side of the known DNA sequence, okay, or the target DNA sequence, and then we essentially put the primer and run the PCR process, and there is a signal growth because of the growth in the chemical signal or growth in the number of copies, I would be able to assertively say that the bacteria X or the pathogen X that I was looking at or looking for is present in the circuits, because the primer was designed for that particular pathogen and it went there and it searched that particular sequence of DNA and it successfully amplified. How that can be possible without that sequence being present here? So this kind of is a yes no answer about the presence or absence of the particular pathogens. There are other protocols which are developed substantially, as there has been a lot of research in this area where there are real time fluorescence based detection techniques which also talks about not only an yes no type of answer about the presence or absence of a species, but also about the intensity or the concentration or the availability of that species of interest. So therefore if a signal is stronger, then definitely it is appropriate to assume that the parent concentration of the DNA strand that we were looking at is a huge number. So there is a proportionality in the concentration of what you are detecting to the signal that you are getting from the system. So that in a nutshell is what primers do. Also DNA polymerase is an enzyme which takes nucleotides from the ambient solution and starts to construct the complementary sequences, and also you do need an adequate supply of these nucleotides or so called DNTPs, deoxyribonuclease triphosphates. Okay, it can be a DATP, that means with an adenine group, a DCTP with a cytosine group, similarly with the guanine group and with a thymine group. So these all four, DATP, DCTP, DGTP and DTTP, are mixed in a certain ratio, and actually a certain equivalent ratio, and this is fixed based on how many molecules you are supposed to copy, and also on an idea as to they should never fall short. Okay, so the reaction should not be limited by the unavailability of these nucleotides. So nucleotides are given in excess almost always in the particular reaction. All right, so therefore we are more or less now kind of acquainted to this PCR process. Now there are certain considerations that we need to investigate for designing any PCR process, okay, and we will be actually doing the designing here subsequent slides. So essentially, again, the denaturation is the process of cleaving the molecule by which the DNA loses its secondary structure. Hybridization, on the other hand, cools the single stranded DNA molecule to lower temperature, let's say about 60 degrees or so, and hybridizes the primer. And extension is essentially, when the incomplete double stranded molecules are extended with the help of an enzyme, the Taq polymerase. The enzyme attaches to the incomplete DNA molecules and replicates the missing complementary bases using available nucleotide called primers. And the extension process occurs at a temperature of about 70 degrees or so. So these three temperature states are very critical, okay, and this essentially can be achieved in a thermal cycler, which is conventionally designed in a manner where there is a huge aluminum block, okay, with various through holes, and essentially this is used as a thermal cycling modality for holding together some vials with the PCR fluid on it. And then what happens is that the block is circulated at different temperature points by using a Peltier cooler and a resistance heater. And one of the earliest forms of PCR were really, PCR is such a rugged process done using just dipping into different solutions at different temperatures. So let's say you have water at 90 degrees, water at again 70 and 60 degrees. So you keep on dipping this vial in different solutions, and PCR would be successfully executed. But the rapidity of diagnostics kind of forced the technology to change towards an automated temperature control system. Now with all this in purview, one important aspect that micro scale devices have to offer, as we have talked earlier, is also the low thermal mass, okay, and also the greater amount of surface area to volume ratio. Heat transfer essentially is again a surface area dependent property, and therefore it is most amenable to assume that in such a case, when there is a thin film, let's say of fluid in a certain chamber, and we are trying to oscillate the temperature or vary the temperature of that particular film of fluid, you basically use or you basically get a better efficiency than rather thermally cycling of volume, okay. Here the surface area is very high over the volume. So you are taking let's say a droplet which was earlier in a vial on the top of a chamber and spreading it into a thin film, and in the process the molecules of that film would be subjected to more amount of heat than all clumping together in a droplet form in a certain vial. So therefore microchips have been designed off and on for doing this activity of polymerase chain reaction, and there has been very many successful models which can generate amplification at a great speed and with great rapidity and accuracy by exploring the microscale or microsystems design and architecture. So some design considerations for the PCR reaction itself is that if you look at time scales of the various cycles, the denaturation, the hybridization or annealing, or the hybridization or the annealing cycle, then the process is really rate limited by the way that the enzyme would be able to stitch the different base pairs and make a complete DNA strand, okay. And so extension process is really the slowest step of the whole PCR process. Denaturation almost always occurs. The primer binding also doesn't take much time, but the extension does take a lot of time, because it is actually the enzyme which goes over a certain base pair, tries to read it, tries to see if there is complementary around it, and then tries to make a hydrogen bond or formulate a bond. So, it is a catalyst, it does some catalysis, it is a catalyst, and then sutures the phosphate group on the sugar, on the last sugar. So, therefore, it kind of zips, first of all makes the zip and slowly starts zipping the DNA. So, that is why it is the slowest step of the process. So, normally the minimum time of extension process is limited by the length of the segment to be amplified. Okay. The speed of the enzyme itself is about 30 to 100 base pair per second, and also the diffusion of primers are important aspect in a PCR reaction. So, therefore, the enzyme can suture only at a rate of about 30 to a maximum of about 100 base pairs per second. So, the maximum speed that the enzyme can actually go and kind of do the suturing action is about 100 base pairs in 1 second. And the way that the primers would diffuse, and essentially all chemical reactions are to a majority or to a major extent limited by the diffusion step or the diffusion rate. So, here we have to ensure that the primer really goes and hits the right molecule which has just been denatured, and it is just ready for annealing the primer onto it, right. So, the primer has to really diffuse through the whole mass, and just because it is a thermally cycling process, there are convective currents which are there in the medium which would help or aid this process of diffusion, but still it does have some rate limitations. Okay. So, that is another aspect. So, there are two aspects, one is the enzyme, the rate at which it sutures, and then another aspect is the diffusion of primers and the other moieties like even the DNTPs or the ionic transport across the solution which maintains a certain pH, so on and so forth. Miniaturization. So, the miniaturization definitely helps in dramatically improving the PCR cycle dynamics, okay, by providing a faster heat transfer rate as we have already talked about, and so therefore, there will be almost always a faster PCR cycle time because of this miniaturization, and the above 3 steps complete a whole PCR cycle. Okay. So, repeating these steps for n number of times would essentially theoretically mean repetition of a single molecule to about a 2 to the power n fold. So, essentially repeating the PCR cycle n times amplifies a single double molecule to a 2 to the power n folds of the same molecule of the same sequence, identical copies. Our question is, does it really happen this way? So, if you look into the way that diffusion kinetics works, that is a major problem. So, therefore, the reaction is slow at the onset, at the initial phase, because the primers are not really diffused to the point where they are supposed to. The DNTPs also are not very well diffused, and so therefore, there is a question of delivery rates of these different moieties to the exact site of their suturement. Okay. And because that is a limitation in real practice, the PCR process is slow at the very outset. It picks up then and after a while, because of the exhaustiveness of the, or because of the exhausting of the concentration of one or more species in the solution, it falls down back again. Okay. So, therefore, in real practice somehow the process is limited by the primer concentration, also the concentration of the DNTPs, and the diffusion rate, okay, by which the molecules transport within the solution. So, there is an important factor here called the amplification factor gamma, which is of some importance. In practice, there exists an amplification factor which is also dependent on the cycle number. Okay. So, normally it is 2 to the power n. So, the amplification efficiency should be 1. So, EPCR n here should be 1, but that is not what happens, and so therefore, this varies also as a function of the number of cycles. So, gamma is equal to 1 plus EPCR n to the power n, where n is the cycle number, okay, and EPCR is the PCR efficiency, which is also a function of the cycle number. So, therefore, we see that for the initial few cycles, let us say for n less than 30 cycles, the EPCR is 1. Okay. This is after assumption that the diffusion has taken place and the molecules are distributed all throughout the matrix of the chemical reaction. And for a number of cycles more than 30 cycles, the EPCR drastically drops down, and this kind of plateaus the growth rate for the DNA of a particular sequence, right. So, therefore, the gamma here 1 plus E PCR efficiency, PCR function of n to the power n would tend to 2 to the power n for n less than 30, and would be much smaller than this 2 to the power n for an n value, so it will be much smaller, 2 to the power n, for a n value more than 30 cycles. All right, so that is what the dynamics of PCR processes. Let's just physically look at some examples. Let's say we want to determine the extension time for a PCR process, and what is essentially given is that the DNA segment to be amplified is about 1000 base pairs long, and the enzyme in use is a Taq polymerase, which has a suturement rate or an extension speed of about 50 base pairs per second. So the time that it would take really for the extension is around 20 seconds. Okay, so time for extension equals the length of the segment divided by the extension speed. That's about 1000 by 20, 1000 by 50, 20 seconds. Okay. Now let's do another example wherein we are suggested that the concentration that is required. Give me a minute here. Okay, so the concentration required for a perfect detection in a DNA assay is about 10 to the power 5 copies. So this is what the final concentration of the DNA assay should be, the PCR reaction should be, and that's per mil of the sample. We need to ascertain how many PCR cycles are needed for an initial sample with a concentration of, let's say, 100 copies per mil, and we had to assume an EPCR of 1. So we really write down this equation: 1 plus EPCR as a function of n to the power n is essentially 10 to the power 5 by 10 to the power 2. 10 to the power 5 is the desired number of copies at the end of the PCR process, and this is the initial sample, EPCR being 1. So this happens to be about 1000, and so therefore the n can be accordingly determined. So here, therefore, n, if n is equal to, or if n were 9, then 2 to the power n would be about 512. If n were 10, it is about 1024. Okay, so essentially it is somewhere between 9 and 10. So we can say that the number of cycles here is about 10. So this concentration would reach in about 10 cycles or 10 thermal cycles for this particular reaction. So I would like to also introduce another very interesting area, that once the DNA is being copied into several different copies, how do you read the signal really on the DNA? Okay, how do you ascertain that the DNA is amplifying? It is in the molecular state. So either you have to get a characteristic spectra, or you have to get some kind of an imprint or a signature which can show you in terms of intensity growth or in terms of some other factor that the DNA is increasing, or the number of copies of DNA is increasing. So there are many detection methods to establish this increase in signal. One of them being fluorescence, and essentially fluorescence is again a physical phenomena wherein there is a tendency of getting a certain orbital configuration in a manner where there is a transition of an electron which takes place from a ground to an excited state, but then the electron comes back in a step. So it actually comes back to a lower excited state first, and then a bunch of different electrons all together come back onto the ground state. And the very fact that it goes from a higher to a lower excited state is nothing but an energy loss, and it is called a phononic loss. It's essentially vibrational loss because of the bond vibrations in a particular molecule. Okay, but at the same time, what is interesting to know is that if such an orbital state is developed, there is a continued high quantum yield emission from a particular agent which would develop essentially those states. In this case, or in the case of the DNA, as you know, the DNA structure is like a twisted ladder. So it is like a cage. Okay, and there are certain fluorophore molecules which kind of rhyme with the size of the the helix on the DNA, so about 2 to 3 nanometers, and they easily go and fit inside this cage. Now they do not bond chemically. The way they go is that they would sit inside the cage, and then there is, because of this presence of so many sp2 sp3 orbitals in the, especially in the base pair regions, there is a tendency of formulation of these ground excited states, and suddenly there is an increase in the quantum yield in terms of emission. Okay, if such a configuration is excited using the excitation frequency of the particular dye of interest, it would emit at a very large rate at a high quantum yield, and that would also determine the number of copies of the DNA molecules which are there within the solution. So fluorescence essentially is the means of determining the signal from which we can also interpret how many DNA molecules have been amplified or copied. What is also important here to mention, for me to tell you, is that fluorescence is definitely a method of ascertaining the real time kinetics of the reaction. So as the PCR process happens, it goes on, and then there is a growth in amplification of the molecules. On a real time basis, the fluorescence spectra does report what is going on. If there is a slow growth in the molecules, it reports a slow growth in the fluorescence. If there is a rapid growth in the molecules, it reports an exponential growth in the fluorescence. Okay, if there is some kind of a plateauing action happening because one or more of the reactants have depleted in the overall reaction and the copies are not getting formulated anymore, the fluorescence signal would also report the same. So it kind of gives a real time snapshot from time to time of the whole PCR reaction. So this is also known as real time PCR or qPCR, quantitative PCR process. Okay, so let us look at some of these different dyes, okay, or how innovatively we can make a combination of the dyes and the primers so that the RT-PCR process can be successfully executed. Okay, so there are several techniques which have been cited off and on in literature, and a lot of research is existing or is going on in this area, wherein they develop different dyes and also different configurations of these dyes or conjugation of these dyes to different constituents of the PCR reaction. So what I can recapitulate, or what we can actually tell at this time, that at this point of time there are about close to 4 different techniques through which fluorescence can be reported. The first technique is just an intercalation. Okay, it is essentially a case where there is a molecule which would just go into a DNA structure and start fluorescing. The second case is a combination of essentially a quencher and a dye molecule, and these are bound to 2 flanks of a primer. This technology is also known as the Tachman PCR, okay, or the Tachman assay. The third case is a molecular beacon, where we talk about a hairpin loop DNA which has a fluorophore and a quencher, and which exactly do the same thing by separating the two and getting a signal. And in the fourth case, there is again some kind of a FRET technique, a fluorescence resonance energy transfer technique, wherein 2 fluorophores of different wavelengths of excitation and emission, and with some connection between the 2, are brought close by. So bringing them apart would change their emission characteristics. So in the next lecture, we would like to discuss some of these aspects and then do some device design for PCR microchips. Thank you.